Good morning, dear students. I am Dr. Salvinder Singh, working as a professor in the Department of Agriculture Biotechnology. And today we are going to take one very important experiment on genetic transformation of crop plants. As you all know that Agrobacterium is one of the most successful and proven method by which we can transfer any foreign gene into the crop plants. And it is a soil-borne bacteria. It is also called a natural genetic engineer because in the natural condition also this Agrobacterium can transfer its own DNA, which we call the tDNA. It also transfer in the wild type, in the wild strains also of Agrobacterium, they transfer their own genome into the crop plant, particularly in the host specific like dicot plants. So, but using those advantage of Agrobacterium as a natural genetic engineer, we are now using Agrobacterium to transfer our own gene of interest into the different crop plants. So today we are going to discuss, or we will demonstrate you how we can produce transgenic banana plants mediated through Agrobacterium using a very important explant which we call embryogenic cell suspension. So we will demonstrate you all the steps of genetic transformation system, right from explant preparation, then how to induce the callus, how to induce the embryogenic calli, then how to establish a embryogenic cell suspension. Then this your embryogenic cell suspension we generally infect with the Agrobacterium. Then we allow the embryogenic cell suspension to undergo maturation to develop mature embryos like somatic embryos, and those somatic embryos which mature, then we put them in the selection. Okay, and in the different selection pressure, here in particular in case of banana transformation, we are using hygromycin as a plant selection marker gene. So, which is one of the most successful or proven your antibiotic selection marker gene, which we use generally in most of the experiments of transgenics. We use hygromycin. Some people also use kanamycin as a selectable marker gene, but whatever the construct we have prepared for genetic transformation for banana, so all our plasmids or the constructs, they contain hygromycin as a selection marker gene. Well, student, today in the whole experiment, what we are going to do today, there is my PhD scholar, Mr. Longkumar. He is the person who is working with me for last 4-5 years, and his expertise in genetic transformation system in banana using the ECS. So he is going to demonstrate you all the whole experiment of genetic transformation in banana. This is the male flower of Bhimkol, which is a diploid seeded variety of Musa balbiciana. Here we use only 11 to 5 brackets of male flower to induce the embryogenic calli, and which shall be used to establish the embryogenic cell suspension.
After we inoculate the male immature flowers into the callus induction media, we will keep it for around 4 to 5 months with repeated subculturing. This one plate is after 3 months of subculturing, as well as this 3 to 4 months after subculturing, after inoculation, which will give rise to our embryogenic callus. The embryogenic calli will be a pale yellowish in color, and this embryogenic calli we are going to initiate the ECS, which we can see over here.
These embryogenic calli inoculated in liquid medium, which is modified MS medium, and maintained under dark in the environmental shaker at 26 degrees centigrade. And it took almost 3 to 4 months to establish the embryogenic cell suspension.
The fine cells that we see here, these are the embryogenic calli which we have cultured in the liquid media. Okay, right now I am preparing for the infection of the ECS. For this we'll be doing through the vacuum method. Now I am placing the filter paper where the cells will be collected once the vacuum is being done, which will remove all the excess media and only the cells will remain on the filter paper. and then we'll transfer the filter paper onto a co-cultivation media and we'll keep it for around 2 days. For this, right now I'm taking my embryogenic cell suspension, which contains my embryogenic cells, my embryogenic calli, and then I'll be putting on top of the filter paper. Okay. This is around 15 ml of my ECS culture, and I will be infecting it with the Agrobacterium, which I have grown and cultured it for 48 hours. Around 5 to 7 ml of my Agrobacterium, and I'll let it infect for around 45 seconds. So after 45 seconds, now I'll switch on the vacuum pump, which will create a pressure deficit where the media will fall to the bottom and only the cells will remain on top of the filter paper. In this way I can isolate and I can dry the embryogenic cells, and then I will be putting it on a co-cultivation media which has been supplemented with acetosyringeone for around 2 days. Once the media is taken out, I'll switch off the vacuum and I will transfer this filter paper to a co-cultivation media, and this plate will be kept in the dark for 2 days, 48 hours to be precise. After co-cultivating the ECS with the Agrobacterium in co-cultivation media for 2 days, the ECS will start to proliferate, and once you transfer them to a maturation media, the embryogenic cells will give rise to somatic embryos, which we can see over here with the white ones, which are proliferating well in the media. And after keeping it in the maturation media for around 2 months and transferring them to the M4 regeneration media, those somatic embryos will start to proliferate and they will shoot out, which we can see over here with the green shoots which are being proliferated, which we can see on the media. Then we further subculture it into the same M4 regeneration media, where we can get multiple shoots from each somatic embryo. Then we're going to put those regenerated plantlets for rooting in a rooting media, and then we're going to grow it for another 1 or 2 months. After that, individual plantlets will be taken and will be hardened and transferred to the greenhouse.
Well, student, we have come to the end of this practical session of today's practical class, and hopefully we explained you all the steps very nicely, and it will be helpful for you to understand what is the genetic transformation of plants is we do to develop the transgenic or to introduce any our alien or gene of interest into the crop plants. And now if you have any questions or queries, yeah, please you can ask me.
Sir, apart from Agrobacterium mediated transformation, is there any other method of transformation in crops?
Well, very good question you have asked. There are actually two methods. I forgot to explain you before. There are two methods of genetic transformation system. One is called direct method and there is another indirect method. So whatever we have demonstrated you today, it is the indirect method, that is called Agrobacterium mediated transformation. through mediated through Agrobacterium. But there is another method called direct method, which we use particle bombardment method, that is a gene gun, where we use a gold particles on which we can coat our gene of interest, either the plasmid or even the your gene of interest with the promoter and terminator, and we can bombard it into the target tissue, which now these gold particles go pierce up to the, sometimes they go up to cytoplasm, some of the gold particle go up to the nucleus, and through eliminate recombination there is a chances of getting we can introduce any foreign gene through particle bombardment also.
Sir, I have a question. Why we are using ECS method for banana genetic transformation? I want to know that.
Well, this is a very good question you have asked. Why we have used ECS as a target tissue for genetic transformation in banana. The advantage of using ECS is that number one, they are completely disease free since we have induced from the male flower, and number two, 1 ml of ECS can give rise to around 1500 to 1800 somatic embryo, and these somatic embryos can give rise to around 80% of these somatic embryos develop the shoots. So from 1 ml you are expecting we will be able to get around 1200 to 1300 transgenic putative transgenic shoots, which is a greatest advantage of using the ECS in our genetic transformation system in the banana. Well, student, this video which we have shown today will definitely help you in learning various different stages of genetic transformation system in banana using the embryogenic cell suspension. And well, this is very much similar to the genetic transformation system in rice also, and where the embryogenic ally is used, but there is a slight modification in different stages as well as in the media composition which we use in banana is different from the rice. But overall, the main theme of development of transgenic rice or banana or chickpea or citrus, all crops, they have the protocol is same with slight modification we have to do. And once we develop any transgenic crop, this is not only going to help our research but also going to help our farmers, because the main purpose of development of GMO, that is a genetically engineered crop plants, is that they are resistant to various biotic and abiotic stresses, which definitely help our farmers. And we are also organizing sort of certified course on various tissue culture techniques for our entrepreneurship development as well as for the students and research scholars. Well, I think whatever the stages or the whole protocol which we have shown you today will definitely help you in formulating your research work, in formulating your research projects. Definitely it will help you in your future academic careers. If you have any queries or your doubts, if you want to ask me anything, please mail me in my email ID mentioned below. Thank you very much. Namaskar.
Hello everyone, I welcome you all to this course on Principle of Plant Biotechnology, and today I'll be taking a practical class. And before that, let me introduce myself. I am Mr. Manav Vikas Gogoi. I am Assistant Professor in the Department of Agriculture Biotechnology, Assam Agriculture University, Jorhat. So in this video, we will show you the preparation of artificial seeds in a laboratory. So before that, let me explain you what are artificial seeds. Artificial seeds are encapsulation of somatic embryo, shoot buds, cell aggregates, or nodal segments of any plant which has the capability to grow into a complete plant when we show the seeds in the soil. One important thing of artificial seed is that artificial seed does not have the seed coat as the natural seeds. So these artificial seeds have artificial seed coats. The concept was originally extended to increase the efficiency of somatic embryogeny or clonal propagation to those species of crops where micropropagation is not economically feasible. Later, the concept of artificial seeds has been extended to increase the frequency of multiple shoot production from axillary buds. Plants has been produced by using artificial seeds of varieties of crops such as rice, maize, and cotton. The availability of high-quality somatic embryos which have the potentiality to convert fast into seedling after encapsulation is the prerequisite for artificial seed production. If axillary bud segments are proposed to be used as an encapsulating material, then we must have a medium which will initiate the regeneration of multiple shoots. Therefore, the procedure of encapsulation of somatic embryo of citrus will be described here. As you can see, these are some of the artificial seeds which we have prepared in the laboratory. So to produce artificial seeds, we need certain materials. As you can see here, this is some of the materials. First of all, we need the X plant. So today we will be using citrus as an X plant. Then we have two important chemicals. One is calcium chloride, another is sodium alginate. So these are two most important chemicals to carry out the production of artificial seed in the laboratory condition. Then we need the liquid medium. Then we have these beakers, as you can see. In these beakers, we will be measuring these chemicals and we will dissolve this. And finally, we need the somatic embryos out of the X plants which we have collected. So these somatic embryos were generated from the X plants which we have collected earlier, and out of this we have generated these somatic embryos as you can see here. And now these somatic embryos which we have generated will be encapsulated by using these chemicals. Now I will describe the protocol step by step. These are somatic embryos of citrus which will be used for today's practical. These somatic embryos of citrus will be bought dried in filter paper. And then we need to prepare sodium alginate 6% in liquid medium. So we'll be measuring 3 grams of sodium alginate. We need to prepare sodium alginate 6% in liquid media. The total volume will be 50 ml. The sodium alginate has to be dissolved properly. Now measure 0.5 gram of calcium chloride dihydrate 75 millimolar using sterile water and make the volume to 50 ml. Now dissolve the somatic embryo which we have generated by using citrus into sodium alginate solution. These somatic embryos are mixed with sodium alginate solution which we have already prepared. The somatic embryos are mixed properly so that it forms popper beads once we drop them in calcium chloride solution. As we have seen that this somatic embryo of citrus has been properly dissolved in sodium alginate solution 6%. So this will be used for preparation of artificial seeds. Put the calcium chloride beaker on the magnetic strider. Hold the pipette perpendicularly. Draw the slurry along with the explant into dropping pipette. Take care to exclude air bubbles. Now hold the pipette perpendicular to the surface of liquid medium containing calcium chloride solution. Then release the dropper slowly such that the alginate slurry comes out the drop with explant entrapped inside them. Here, care should be taken to include only one explant inside one drop. The liquid should be stirred at 45 rpm all the while the dropping is done. As the drop falls into the calcium chloride solution containing the medium, ion exchange reaction occur in which calcium replaces sodium ions, due to which surface complex occur. This gradually hardens the drop into beads. Keep the beads in the same solution for 45 minutes for proper beads formation. Now transfer the beads into a Petri plate. Now as you can see, these are the artificial beads, and these are known as artificial seeds. Now these will be washed with MS Basel medium and blot dry by using a filter paper. So MS medium is being used to wash. Now these beads are blot dried using a filter paper. Now artificial seeds are ready. Properly formed beads will be firm to touch and will not stick to each other. One can plant the seeds in different types of substrate such as agar gel medium at 0.75% and sterilized soil. Now let me brief you about the advantages of artificial seeds. Artificial seeds are high volume, large scale propagation method, maintain genetic uniformity of plants, then one can directly deliver the propagules to the field, low cost of production per plantlet, rapid multiplication can be done, and easier to transport long distance as the size of the seeds are very small in comparison to other seeds. Dear viewers, if you have liked the video on artificial seed production, so please give your feedback in the comment box below. Thank you so much.